We're going to keep talking about human body tissues, and we'll be honing in on epithelial tissue today. Recall that we are in our histology unit, which is the study of tissues. The human body is composed of over 50 trillion cells, and those 50 trillion cells all come together to form only 4 different types of tissues. And then those 4 tissue types will come together to form our organs, and then our organs will come together to form the organ systems. Organ systems will come together to form the organism. So all 11 body systems are made out of only 4 types of tissues, which we introduced at the last lesson. We talked about muscular tissue. We mentioned connective tissue, nervous tissue, and epithelial tissue. So of these 4 tissue types, we are going to focus in on epithelial tissue today. So we're going to be talking about those tissues that make up the linings and the boundaries between the inside of our bodies and the external environment. So we are going to hone in on the characteristics and functions of epithelial tissue. Some of the things we're going to talk about are describing the properties that distinguish epithelium from other tissues like connective tissue and muscular tissue. We're also going to classify the 8 different types of epithelium, and we're going to distinguish them from one another. And then we'll talk about where each of these tissues can be found. We're also going to explain how structure determines function. So of these 8 different types, there will be a different structure to that epithelial tissue, which allows it to have a different function, which is why we find it in different places of the body. And then we are going to try to make it so you can visually recognize each of these tissue types, not just in nice cartoony pictures, but in actual specimens and photographs. Epithelial tissues are definitely widespread throughout the body, but they are not the most abundant. That's connective tissue. So these epithelial tissues are sheets of very closely packed cells. These cells are basically stuck together, and we'll talk about some of those features that adhere these cells together in a future lesson. They are typically going to be either one cell thick or many cells thick, and one of those layers of cells will be exposed to the environment, whether that is the environment on the outside of our body or the environment that makes up some internal space like the lumen or opening of the gastrointestinal tract, or even like the inside of our blood vessels. So epithelial tissue is going to cover most of the body surfaces and lines all of our body cavities, right? So the thoracic cavity, the abdominal pelvic cavity. It's also going to constitute most of our glands. And this tissue is avascular. A means without, vascular indicates blood vessels. So these tissues do not have access to a blood vessel themselves. They rely on diffusion of nutrients from blood vessels that are found in the underlying layers that's typically a connective tissue. So that's how it's going to get its nourishment, is the diffusion of those nutrients from the blood vessels within the connective tissue. And everything that enters into the body or leaves the human body is going to have to pass through one of these really tightly adhering layers of cells, or an epithelial layer. So when it comes to function, think about forming boundaries between our body and the external environment. So there's lots of boundaries, right? There's boundaries within your eyes. There's boundaries with your nose, within your mouth. Even the internal walls of the respiratory tract need to be lined with epithelial tissue because there's exposure to the air. That's just a tube that goes from your mouth and into your lungs. So there's going to be different structure of this epithelial tissue depending on where we're looking at in the body. And so that structure is going to provide different functions. In areas where we need to control substances leaving the body, that is going to have a layer of epithelial tissue that has an anatomy focused for filtration. If we're thinking about like glands, we have glands that need to produce hormones that are vital for our body functions, or mucus or sweat to cool our bodies down. So these cells will have a different structure or anatomy that allows them to be really good at the secretion of substances. Some of the epithelial tissue will have many, many layers, and that is going to provide protection of deeper tissues from injury. We're also going to have specialized nerve endings in epithelia that allows us to detect a stimuli, so it provides for sensation. So we have functions like protection, secretion, excretion, absorption, filtration, and sensation. That's all going to be determined by the anatomy of that epithelial tissue. No matter which of the 8 epithelial tissues we're talking about, there is going to be some structures and some characteristics that all 8 epithelial tissues share. So some of these general structures are that these cells are going to be very close together. So in drawings, usually you're going to see those cells drawn right next to each other. There's no space in between those cells, so there is a very small amount of extracellular material. These cells are also avascular, meaning they do not have blood vessels. So there's going to be an underlying connective tissue down here that is going to contain these blood vessels. And so those blood vessels are what's carrying the oxygen and sugar or hormones or whatever nutrient we're talking about here. And then those nutrients will rely on diffusion to leave the blood vessel, and then they will slowly move into that epithelial tissue. So it can be used by those cells. These cells are also going to have a high mitotic index or high rate of mitosis. And typically the cells that are going to be doing mitosis are going to be found in the bottom layer of the tissue because that's where the nutrients are going to be coming from that underlying connective tissue layer. So only the cells that are close to those nutrients are going to get the nutrients that they need in order to make more cells. The cells that are too far away are going to typically die. So we'll have a high rate of mitosis in the cells that are near that connective tissue with all of the blood vessels. And so between the connective tissue and the epithelial tissue, there is going to be what's called the basement membrane. And that basement membrane is like a glue that's made out of this collagen and glycoproteins and other protein carbohydrate complexes that will anchor or glue that epithelial tissue to the connective tissue that's down below it. So the basement membrane is like that glue. We're also going to have the opposite side, which is exposed to the external environment. So if it's the skin, it's that upper layer of the skin. If it is inside of a gland, like a sweat gland, or maybe it's in your gastrointestinal tract lining, that exposure to the innermost part of the tube is called the free surface. The free surface is not touching any other cell. And then the bottom, just above the basement membrane, on that bottom layer of cells, that basal layer of cells, there is the basal surface, which is right on the bottom. So that would be this area right here, in between the basement membrane and the nucleus of the cell. It's like the bottom part of the cell here. The way that we classify epithelial tissue is going to be by the number of layers and by the shape of the cell. And the number of layers and the shape of the cell is the anatomy, and that is going to determine the function. So the free surface is also called the apical surface. So it is the surface that is not touching any other cell. It faces away from the basement membrane. The basal surface is the surface that is facing the basement membrane and comes into contact with it. And then we're going to have a lateral surface, which is between the basal and the apical surface. It's like the sides or like the wall of the cell. And when we have one layer of the cells that's anchored to the basement membrane, that is said to be a simple epithelia. And then if we have two or more layers, that is called stratified epithelia. Right? So not all are going to come into contact with the basement membrane, just that bottom layer of cells. So here we have some pictures. There is the simple epithelium. So we've got just the one layer of cells versus the stratified, right? So if you look at these, you have the free surface or the apical surface, which would be this here. In the stratified side, it would be this layer here, right? And then we have the basement membrane, which would be this kind of dark orange line, right? This dark orange line, and then that lighter kind of speckled structure, that is the connective tissue that's down below. And then we're going to talk about what's called pseudostratified, and those pseudostratified, they look like there's more than one layer, so they look like they're stratified, but they're actually not. They're little tricksters, and they are really actually one layer, but they are these tall column-like cells, and they are falsely stratified or pseudo. And then we have the shapes. So the shapes can be squamous or squamose. I think of them as being squished, and they kind of look like a fried egg there. So they're definitely going to be longer than they are tall. There's cuboidal, where they're square or round-ish, because we're not plant cells, so we don't have cell walls. So our cells really aren't square. They're more of like a round, but they're going to be just about as tall as they are wide. And then there is columnar. With the columnar cells, these are going to be tall like columns, and they're taller than they are wide. So all of the cells, whether they are columnar, squamous, or cuboidal, these cells are going to be really tightly packed together with very little extracellular matrix or inter means in between. Because these tissues form coverings and linings, right? So we're trying to make a boundary. And because they are forming those coverings and linings, there's going to be one free surface that does not come into contact with the other cell. So that is that apical surface. And then opposite to that free surface, the cells are going to be attached to that underlying connective tissue by a basement membrane. So when it comes to our epithelial tissue, we have two categories. There is the covering and lining epithelium, and then we have glandular epithelium. So these cells are going to change as they go into what's called the secrety units, right? So these are deep within the gland, and these are the guys that are actively secreting the hormone or the sweat or whatever substance it is that they're made to secrete. And then that substance will either be deposited directly into the bloodstream, or it might fill a duct. And that duct will go to the apical surface of the cell. So these cells here, these would be the lining epithelium. And then these deeper cells that are inside of the duct, that would be the glandular epithelium. So the covering and lining epithelium, that would include the epidermis or the lining of the blood vessels and ducts, the lining of your respiratory, reproductive tracts, while the glandular epithelium, that is the secreting portion of the glands, right? So like your sweat glands, the ones that are actually producing sweat. And then it will also include the thyroid and adrenal glands, which are endocrine glands. And they're going to secrete their substance instead of into a duct, it will go into the bloodstream. So let's first talk about the classification of the coverings and lining epithelial cells. So remember that we're going to classify these tissues in two ways. We're going to look at the number of cells, and then we're going to look at the shape of the cells. So our simple epithelial tissue is going to only be one layer, while the stratified is going to be more than one layer, so two or more. And so when we're looking at this simple epithelium, if it has one layer of cells, those cells can be one of these three shapes. If they are squamous, they are these thin, scaly-like cells that look like they're kind of squished. They look kind of like floor plates or like ceiling tiles. The simple cuboidal is going to be more of a roundish shaped cell, and it's going to be equally as tall as it is wide, where the columnar cells are going to be these really tall and narrow cells, right? They're going to be taller than they are wide, just like a column. And then there is what's called transitional epithelium, and we have pseudostratified columnar epithelium. So with the pseudostratified columnar, not all of the cells are going to reach the surface, but they all reach the basement membrane. So it is one layer. They just don't all get up to the top. And so as a result of that, the nuclei are not going to be nicely lined up, and it's going to appear to be stratified, but it's just because some of the cells are taller than the other cells. And then we're going to see in a simple columnar epithelium, we're going to find the addition of goblet cells. They get their name because they are shaped like the cup, the goblet. It's like a wine glass kind of shape that secretes mucus. These guys are mucus secreting machines, and that's all they do. So we're going to find them in our mucous membranes, right? So your mucous membranes are going to be made up of a simple columnar epithelium or a pseudostratified epithelium, because those are the two epithelium that we find these goblet cells in. So if we look at simple squamous epithelium, this is a single flat layer of cells. It's very thin, which if you think about diffusion of nutrients, right? If you need the rapid diffusion of gases, that would be a great tissue to have in that location. So molecules will move from an area of high concentration to an area of low concentration, right? And so if you think about like in the lungs, you need rapid diffusion of oxygen into the body because the heart isn't going to stop pumping. So blood's going to keep moving. And you need to have that oxygen get delivered into the blood very quickly. You also need the carbon dioxide to be able to be removed from the body very quickly. So by having this thin layer of cells, you're going to see the rapid diffusion of things like oxygen, right? We'll be able to go from an area of high concentration in the alveoli of the lungs into the blood vessels where there's a low concentration of oxygen, where carbon dioxide, where you have a lot of it in your lungs because you're trying to get rid of that waste product. If it has a thin layer of cells to get through, it's going to be able to diffuse very quickly out of the blood and into the alveoli of the lungs, and you can exhale. So we're going to find this in places like the lungs or the blood vessels where you need to have a rapid diffusion of materials. If these were really tall, thin, tall cells, it would take too much time for that diffusion to happen and you wouldn't get enough gas exchange in the lungs and in the tissues. So if you were standing on the apical surface, so you're looking down at a simple squamous epithelium, this is about what you would see. These cells are very tightly packed together, right? Because we're forming a barrier. And they are kind of squished. So this is as if you're standing on the free surface and you're looking down. Well, here what you see, this is as if you are looking at a cross section, right? So here we actually have some glandular tissue right here. These are secreting units of a gland. So all of this, so kind of ignore that for right now. But if you look at this right here, this is open space, right? There's no cells here, right? And so what we're actually looking at is a serosa. And so this would be the free space, right? And it's as if you are looking at one of these ducts and it's just cut the other way, right? So it's a longitudinally cut. So the free surface or apical surface of these cells are here, right? So this is the free surface right here. That's the apical surface. Your basement membrane is the opposite side, so that's actually here. So this is where that layer is getting glued to the layer down below. I know this is kind of probably throwing you through a loop. This little guy is actually just doing mitosis. So he's just making it kind of more difficult for us, but there we go. So there's your little basement membrane. And then we've got the basal surface of this epithelial tissue. So this right here would be the basal surface, okay? So we have to kind of use our imagination here to draw in the actual individual cells. But I feel like most of you guys can see the nuclei, right? So here I have a nucleus, here is a nucleus, here is a nucleus, here's another nucleus, right? So all these dark purple portions, those are the nuclei, right? And so the cell membrane, you have to use a little bit of imagination. But I'm just going to outline this guy here. So there's one cell. Here's another cell. Here's another one. Oops. Let me redraw that one. And as I'm drawing them in, if you look at the shape of what I'm drawing, I hope you can see that these cells are definitely longer than they are tall. So they're kind of squished, giving that squamous shape. And there's one layer of these cells, right? So this is a simple squamous epithelial tissue. Here I have a picture of simple squamous epithelium in the alveoli of the lungs. And so you can definitely see the nuclei, right? These little dark purples are the nuclei. And then these cells, so that's a little cell there, right? Here's another little cell. You just can't see the nucleus. There's another cell. Here's another cell. So this is one layer of these thin cells, and it's going to permit that rapid diffusion of substances. There's actually a really good assignment that we're doing. It's one of the APR Module 3 tissue histologies, where you actually get to look at the simple squamous epithelial tissue. And so the alveoli, or the sacs of the lungs, are these spaces in between. So here's an alveoli, here's an alveoli, here's an alveoli, right? So that's where the gas exchange is going to happen. And what you see right here, this is a blood vessel, right? And so the gas is going to go through these alveoli and into that blood vessel. When it comes to simple cuboidal epithelium, this is going to be, again, a single layer because it's simple, but these are going to be cube-shaped or roundish cells. These guys are going to function for the secretion and absorption of substances. So, for example, in the kidneys act as a filter in your body, and they filter your blood like 300 times a day. So these cells are going to absorb things from your blood, and then they will secrete that certain substance into a duct, which will then collect and form your urine. So they will have to absorb those things from the blood and then secrete it out into another tube. We're also going to find this type of epithelium anywhere along a duct. So if you have to choose any single function, focus on the secretion, because you're going to find this lining like your sweat glands and lining your endocrine glands that are making hormones, and they have to secrete those hormones into the blood. Here is a sectional view of a simple cuboidal epithelium, specifically within the kidney tubules. So remember, this is as if a long section was cut through, right? So it's a longitudinal section. And so you can see that the free surface would be this portion right here, right? That would be the free surface area of the cells. And then you have the basal surface, and that basal surface would be here, this bottom part of the cells, right? That would be the basal. And then the basement membrane is, you can actually see the basement membrane pretty good. It's this dark blue. That's kind of acting as the glue there. You can see that basement membrane connecting these two layers. Okay, so that's separating there. And so you have one layer of these cells that are just about as tall as they are wide, right? And then this would be the duct. So they're actually releasing their little substance out into this open space, right? And so you can actually see some of the substances being released like here and here. And then if you were to take those kidney tubules and cut into them the other way, so we would do a cross section, right? So we're cutting through the shorter way, and it's as if you're looking through like the tube of a straw. What I hope you can start to notice are something that looks like donuts. See these circles? They look like donuts to me. So if you look at these circles, each one of these sets of circles represents a simple cuboidal epithelium. So you're going to have your free surface that instead now is going to be on the inside part of that circle. Right, so this is the apical surface or free surface here. Boop. Right, and when they release their substance is going to be released into that open space. And then you can definitely notice the nuclei, at least I hope. So that's this purple kind of darker stain. That's the nucleus of each cell. And then you kind of have to imagine the cell membrane in between those nuclei. And once you get those cell membranes imagined in there, I hope you can see that these cells are about as tall as they are wide. So it's a nice cuboidal epithelial tissue. And then the basement membrane, since we're all about these, so then I got a basement membrane here, outside, right? And then here's a basement membrane here, and then a basement membrane here. Right? So the basement membrane is like the outside part of the donut, and then the apical surface is the inner part of the donut. So if you have to identify the basal surface, right, the basal surface would be this part in between the nucleus and that basement membrane. There. So our simple cuboidal epithelium is going to be this single layer of square-ish, round-ish cells, and they are going to focus on the absorption and secretion of substances. So anywhere you see secretion, you should be thinking of these cuboidal epithelial tissues. So like in the kidneys and in our saliva, salivary glands. If those cells that you're looking at are taller than they are wide, and we have this single layer of tall, closely packed cells, we are looking at simple columnar epithelium. And so we're really going to find these along the lining of the digestive tract. And some of these cells are going to have these little hair-like extensions called cilia. Some won't. And others will have those goblet cells that we talked about that secrete mucus embedded in between the columnar cells. But typically what you're going to see is those nuclei will be nice and lined up, and each cell will reach up to the apical surface and the basement membrane. So when we have these columnar epithelium, we're going to have the secretion of mucus, if there's those goblet cells, and absorption. So in the small intestines, we're going to find this simple columnar epithelium because that's where we're absorbing our nutrients. And so these guys are going to be able to grab those nutrients, and those nutrients can then get processed in any way it needs to be before it is absorbed by the blood and used by the body. So think of absorption for the simple columnar epithelium. And here I have a longitudinal section of the gastrointestinal tract here. So this is where like the food would pass through, right? So this is the empty space, which means the free surface of these cells is going to be this layer here. Right? So that's the free surface. And you can see these little tiny wispy projections. That's the cilia. Right? So now I have cilia here. Cilia. No, the guy from Khan Academy does it. Okay, cilia. So then you have your basement membrane, and that basement membrane is going to connect this tissue to the underlying connective tissue. So this is when your keen eye is going to have to come into play. The epithelial tissue typically stains a darker purple. And I hope that you can see a difference between these cells and all of these cells. So all of this here, this is all of the all of the connective tissue, right? That's all connective tissue. So that's not where we want to be right now. Right, we want to be up in the epithelial tissue, which is by that free surface, right? So we're up here. So my basement membrane is going to be that line between the connective tissue and this epithelial tissue. So right here, this little line. And then the basal surface of this epithelial tissue would be here. Right, all of this would be the basal surface, right in between that basement membrane and these nuclei here. Then you can imagine those cell membranes, and you'll see that those cells are definitely taller than they are wide. So I have this nice, simple columnar epithelium. And then actually hidden in this simple columnar epithelium is a little goblet cell. So if you look right here, here is a cup-shaped cell or a wine glass-shaped cell. And that little white drop, that is actually a droplet of mucus that he is releasing into the lumen here, that opening. So if you have a single row of these tall, narrow cells, we're looking at simple columnar epithelium. And some of it will have that cilia micro border. So we also might find these goblet cells that secrete mucus. So this is actually a really good goblet cell here. And I would never make you identify this as the goblet cell because he looks a lot like the simple columnar epithelium cells. If I want you to identify a goblet cell, I would point to this big old droplet of mucus. And I'd say, what is this? And then you'd say, that's a goblet cell. So, you can also see that little brush border here. It's got those cilia. And we're going to find this in the lining of the GI tract. We're going to find it in our uterus, kidneys, and even the uterine tubes. So pseudostratified is a type of columnar epithelium. And if you want to try to identify the pseudostratified, you're going to look at the location of the nuclei. The nuclei are going to be found at different levels, so it appears to be stratified, but it's actually not. And these guys typically have goblet cells and they have cilia. So they're going to be secreting a bunch of mucus, right? So secretion. And then that mucus can capture particles, and the cilia can beat and move those particles up so that way they don't go down into your respiratory tract. Underneath a microscope, I always just call these nuclei. I say like it's a hot mess express in here. These nuclei are all over the place, and it looks like there is more than one layer, but if you follow very carefully, every one of these cells, even like this guy, he does contact the basement membrane. So we're going to find this only really in the respiratory tract. So that way those goblet cells secrete the mucus, it can capture any particle, and then the cilia will brush and beat, moving the captured substance out of the body. And we're also going to find it in portions of the male urethra. Urothelium is also known as transitional epithelium. And so I'm trying to update my vocabulary because I was taught transitional epithelium, but your book uses urothelium. So with this urothelium, there is two different looks depending on if the organ is full or empty. So there is still a free surface of the cell, but it's going to vary in appearance based on how much it's stretched. So we're going to find this in like the lining of the urinary bladder. Right? We're also going to find transitional epithelium in the uterus. But when it is squished, that's because the bladder is full or the organ is full. So as the organ starts to fill up, it's going to push up on that basement membrane, and it's going to push the basement membrane closer to the free surface, which is going to squish those cells together. So when the bladder is full, it's going to look like it's a stratified squamous epithelium. But the key to identify this urotransitional epithelium is to look at that free surface, particularly when it is not full or distended. So if you look at the free surface, you'll see that they kind of look like clouds. They go up and they are rounded. Right? So that round portion at the free surface, that is what's going to tell you that it is this urothelium or transitional epithelium. And that ability to stretch and be squished is going to allow for filling of the urinary tract. So we're also going to find this extended up into the ureters. So the urothelium is unique to the urinary tract, and we previously called it transitional epithelium. So that is technically stratified. Okay, stratified epithelium ranges anywhere from 2 to 20 or more layers of cells. I mean, you can have like 40, 50 layers of cells. Some of these cells will rest directly on others. And only the deepest layer, typically referred to as the basal layer, is what attaches to the basement membrane. And we're going to name these strata based off of the shapes of the cells, just like we did before. So we have stratified squamous, stratified cuboidal, and stratified columnar. Stratified columnar epithelium will have many layers, and the free surface is lined with columnar cells. So when we're identifying stratified epithelium, we're always going to look at the apical surface or the free surface. Okay, so this free surface is lined with columnar cells, and these are very rare in the body. We really only find this in your larynx, which is like where your voice box is, leading into the trachea, and in the male urethra. And so typically the deeper layers are honestly more cuboidal in shape. Stratified squamous epithelium, we're going to do the same thing. We are going to look at the apical surface or the free surface to identify which type of epithelium it is. So if you look at it, the apical surface, those cells are squished. They are longer than they are tall, so they are squamous in shape. Even though as you get down towards the free surface, or the basement membrane, I'm sorry, you might be saying like, hey, Murphy, those are kind of cuboidal in shape. You are right. Down at the basement membrane, those cells are actively doing mitosis, and they typically are a cuboidal. But remember, we're going to look at the apical surface, so this upper surface, to identify stratified epithelium. And so we're going to find this stratified squamous epithelium in places where we need protection, because it's going to provide thick layers so we can protect those underlying tissues. So think about like the epidermis of your skin. We need to protect all the underlying layers, and so it's pretty beneficial to have some nice, thick, stratified squamous epithelium. So if there is something like a thorn that you step on and it penetrates into some of those layers, you still have more layers allowing for protection. Or if you have ever not fully chewed a tortilla chip and you gave a nice swallow and you swallowed that tortilla chip and maybe it's cut the roof of your mouth, or even, as you swallow, it hurts. And if you had one layer of cells, that tortilla chip would completely demolish that thin layer of tissue and it could cause hemorrhaging. But it's made out of stratified squamous. So even as you swallow that tortilla chip, if it scratches your esophagus, we're still going to have many layers that can still provide for protection of any of the structures that are underneath that layer. So that stratified columnar epithelium is rare, and we're only going to find it in places where like two different epithelial tissues meet. So it's kind of like this bridge in between. And then if you think about stratified squamous epithelium being what makes up the epidermis of your skin, it's pretty widespread. Right? And typically the deeper layers are going to be like a cuboidal, but that's because those are the cells that are closest to the blood supply. Therefore, they're going to get enough nutrients so they can do mitosis. And they're going to have the active stem cells that can do mitosis and replace these old and dying cells. Right? So these cells are located down at the bottom. And as they do mitosis, they are going to push the cells up towards the free surface. And so as those cells get pushed up to the surface, they're going to become flatter as they migrate upward. And they're going to start to die because they are getting too far away from that nutrient supply and the blood supply. And so when they start to die, they will eventually flake off. And so if you think about when our skin gets kind of ashy and we have to do that exfoliation, we're removing that outermost layer of dead skin cells. That happens throughout our stratified squamous epithelium. And when that outer layer of dead cells does flake off, we can see that in a microscope, and it is called desquamation. As these cells get pushed up and migrate upward, they're not only getting cut off from blood supply, they're also starting to become keratinized. And that is from a protein called keratin. And that keratin makes our skin waterproof. It makes it abrasion resistant. It's a really hard protein. And that protein starts to replace the cytoplasm inside of the cell. So not only as these cells are pushed up do they get cut off from their nutrient supply, but they're also going to lose their cytoplasm to this hardened protein of keratin. It's the same protein that makes our hair strong and our nails so strong. And so that keratin replacing the cytoplasm prevents these cells from being able to do all of those metabolism and chemical reactions. And so that's a second thing that's going to start to kill the cells. If the cells are not keratinized, those are typically not going to be at the surface. They are going to lack that surface layer of dead cells, and they're going to form more of the lining of our internal spaces. So like the visceral pericardium or the parietal pleura of the lungs. If we look at these microscopic images, I want you to focus on this area here. Okay, so we have these really tightly packed cells here and in figure B here. right? These are really tightly packed cells, right? So that's the epithelial tissue. This lighter pink stuff down below, that's all connective tissue, okay? And so we're actually looking at plantar skin. So this is the skin on the bottom of the foot, right? So this is a stratified squamous epithelial tissue. So we have the basement membrane. You can see the basement membrane is where there is a line between the connective tissue and the epithelial tissue. So this here. Okay, so this is the basement membrane right here, that little line, right? On figure B, it's here. Boop. Right, so it's this line. And then you have the free surface or the apical surface, which is here. Here. So the cells that are near that basement membrane, they are closest to the blood vessels, right? Because remember, blood vessels are in this connective tissue here, right? So they're like bloop here. Okay, over here, they would be like here. So only these cells that are by that basement membrane are going to be close enough to get all of those nutrients that they need and to get rid of their waste product. So these are the cells that are doing mitosis, right? And as they do mitosis, they're going to push up the cells, and those cells are going to get further and further away from that blood supply. As they get pushed up, they're also going to start to flatten. So they're going to become kind of scaly. And then you can see as they get far from that nutrient supply, these are now dead. And this is the slushing off. If you look right here, these cells are starting to detach from the surface and they're going to slush off in desquamation. As these cells also get pushed off, they're starting to fill up with keratin. And so we'll talk all about these different layers in our next unit, but as they fill up, they're going to lose their cytoplasm and fill up with this protein called keratin. And that is going to cause them to be hardened and resist penetration caused by pathogens, or even if you step on a thorn or something, there's going to be so many layers that nothing's going to be able to get past them. Okay. It's also going to start to become waterproof. And we are going to retain water because of those hardened keratinized cells. Non-keratinized stratified squamous epithelium is going to be the same as the keratinized, but the cells at the surface, these guys are not dead. These guys still are going to resist abrasion and penetration of pathogens. But as these cells fall off, that's when they are dying. So we're going to find these guys in places like our oral mucosa or our tongue, the esophagus. And so if you look at this one, I'll draw out the basement membrane for you like I did the last time. So here we have the color difference, right? There's that darker purple here versus like this kind of spacious cells. There's all that extra space, but if you look here, there's no space between those cells, right? So this is that basement membrane. Oops. And then the free surface would be this portion here, the apical surface there. And so the cells that are doing mitosis are going to be these cells right along that basement membrane. The stratified cuboidal epithelium is going to be very rare in the body, but it's where we have more than one layer of these cuboidal shaped cells. So think about in places where you need a lot of secretion, like your sweat glands, right? You're producing a lot of sweat. So sometimes one layer of these secreting cells isn't enough. You need a backup layer. So we're going to have more than one layer. It is typically two. So these surface cells will be squarish or roundish in shape. And we're going to find them in the sweat glands, so where we need sweat secretion, in the ovarian follicles. Those are structures that produce ovarian hormones. And in the seminiferous tubules, that is a location in the testes where sperm goes to maturate. And once they are mature, they will be released into the duct of that tube and they will continue their pathway. So if you're looking at the cuboidal epithelium, a lot of times that basal surface or layer will actually be kind of squished. So like if you look at these cells here, you have your basement membrane, right, which is here. This is the basement membrane. Oh, my circles are getting awful. And then the free surface or apical surface is here, the inner part of the donut. And you can definitely see that there are these squished or squarish shaped cells, but what you might not notice are these cells here. So that's actually a cell layer. That's a cell layer. And so there are actually, there's a second layer. See, here's another guy there. He's kind of squished. Here's another one. So there's actually two layers of cells here. This actually is a much better picture here. If you look at this one, you got one layer and then you have a second layer right there. So there's two layers of these cuboidal shaped cells. And so you guys should try to think about what are the benefits of these different tissue types and where would we benefit in the body of having that type of tissue, right? So for example, being a simple epithelium, like a simple squamous epithelium, versus being a stratified squamous epithelium, right? Simple, we're going to have that nice rapid diffusion because it's not as much space that the molecules have to go past. And stratified, we're going to have protection. So I'm going to stop it here because this was a really long one and I'm so sorry for that. And we'll talk about glandular epithelium next.